Hey, good morning. It's Wednesday, and I'm Dr. Steve Stites, Chief Medical Officer at the University of Kansas Health System, and this is Show Me the Science, and I'm so excited. If you're a regular viewer, you always know these two things. We're always going to follow the science, and I am the biggest Star Trek fan on the planet. Okay, maybe not the biggest, but I am right up at the top. I'm so excited about today's Open Mics topic because it combines two of my favorite things, medical research and the final frontier, outer space. Yes, today we're going to go to that final frontier with scientists from here at the University of Kansas Cancer Center and the Moffitt Cancer Center in Tampa, Florida. Our panel of experts includes the University of Kansas Medical Center professor and researcher in the Department of Molecular and Integrative Physiology, Dr. Lane Christensen. Joining us virtually from Tampa are researchers Dr. Derek Duckett and Dr. Patsy McDonald. Dr. Duckett is also chair of Moffitt's Drug Discovery Department, and Dr. McDonald is an associate professor of Moffitt's Cancer Physiology Department. Thank you all for being with us. This is going to be a hoot. We're going to have a lot of fun today. We're also going to hear from Dr. Dana Hawkinson, Medical Director of Infection Prevention and Control, shortly when we get to the COVID count. But before we begin our discussion, it's time to define some of the vocabulary terms you may hear our experts use today. Our first term today is surface receptor signaling. Now that sounds like a whole lot of fun out there, something about a train, I don't know, which Dr. McDonald and Dr. Duckett are considered pioneers in this field. Guys, help our viewers better understand surface receptor signaling and why you all dedicate research to this.
So cell surface receptors are receptors or proteins that are found embedded in the plasma membrane or the outer membrane of the cell. They're actually comprised of three domains: an extracellular domain, so this is the piece of the protein that is exposed on the surface of the cell, a transmembrane domain, which is how the protein traverses the cell membrane, and an intracellular domain, which allows the receptor to communicate to other signaling molecules inside the cell. So receptors receive signals from outside the cell, and these signaling molecules are known as ligands. And when a ligand binds to the receptor, it induces a conformational change or a change in shape of that receptor. And that change in shape is what allows the receptor to then communicate to other molecules inside the cell, allowing the cell to respond to that original stimulus or signaling molecule.
So these receptors, guys, they're getting some signal. They tell the inside of the cell what to do, or at least transmits that message inside the cell. Is that kind of it?
Exactly it, yes.
Yeah, it's like when my kids were younger, they'd say, "Dad, I need some money," and then I had to reach in my wallet and give them money. So my kid was the cell, my wallet was the cell surface receptor, I think is what that means. Okay, that's good. Thank you. All right, so let's move to the next question. As we talk most about your recent space experiment, we will hear the term beta-arrestin 1. Dr. Duckett, talk to us about what beta-arrestin 1 means.
So beta-arrestin 1 is also a protein. It's an intracellular protein, and Patsy's the real expert on beta-arrestins, so she'll keep me right if I say anything wrong. But the beta-arrestins are really part of the signaling mechanisms for cell surface receptor proteins called G-protein coupled receptors. And in a nutshell, beta-arrestins are, when they were initially discovered, were shown to desensitize the receptor signaling. So once they're activated by a ligand, you don't want that cell's surface receptor continuously signaling and asking you for money. So you want to turn that receptor off. And beta-arrestins were there to turn the receptor off. But in addition to turning the receptor off, Patsy actually, her work demonstrated that they're also signaling intracellular signaling molecules in their own right. So they act as adapter molecules to scaffold and help other proteins be activated within the cell. So for example, they scaffold MAP kinase and activate MAP kinases, but they also scaffold E3 ligase components and degrade proteins within the cell. So they're regulators in their own right. So that's what beta-arrestins are.
So really,
And I
this is
can
my
just-
wife telling my kids to stop taking the money out of my wallet, but then giving me a honey-do list that I have to still do. Is that what you're saying to me?
That's it.
That's a
Yeah.
great analogy. So
I've
I
got
will just
science
add that. We'll just add that this arrestin molecule gets its name, as Derek mentioned, they were first discovered as proteins that could turn off the receptor signaling. So they arrest that signaling, and this is where the name comes from.
Yeah, that is, okay, I just have to say, I grew up in medical school when we didn't know all this stuff, and trying to read and learn about it afterwards. This is really cool. I mean, this is pretty cool. I say space is the final frontier, but this cellular research, and to think how well and how incredibly designed cells are, that is really complex. Okay, so let's take something that may be a little easier. Microgravity. Is that when I can't get out of bed in the morning, or what exactly, Dr. McDonald, is microgravity?
So microgravity is synonymous, if you like, with weightlessness. And it's where there is close to zero gravity or gravitational pull on the body or any entity that is in microgravity. So it's not zero gravity, but it's so close to zero gravity, it's very difficult to measure, which is why it has the name microgravity.
Okay, I get that one. That is not me going up for a jump shot. That may have been Michael Jordan, certainly not me. All right, so that this microgravity, I have a feeling we're not just talking about these terms randomly. We're going to pull this together. But first, we're going to talk a little bit about a linear accelerator that is used to fight cancer tumors. Guys, how is that used in space research? Why does that matter?
So that's a good question, and probably not one for us, I'm afraid. Linear accelerators, as you probably know, they're used in cancer research so that you can direct radiation towards a particular tissue, the tumor, and avoid hitting normal tissue or damaging normal tissue around that tumor. Exactly how these linear accelerators are used in space, I'm not.
I think we're going to talk about that. Go ahead, Dr. Christian.
So linear accelerator that we use on the ground is based out of the Brookhaven National Laboratories in New York. And that linear accelerator actually activates and sends to tissues heavy protons and large heavy ions, which is not the same as like your X-ray and your radiation proton therapy tools that you use here at the university.
Okay, so this is yet another research tool that we're going to use to study the effects of outer space then. Is that kind of it? Because we know there's all sorts of radiation floating around in space that we have to figure out a little bit more if we're ever going to really get into space, and Lord knows we all want to get out to space. Okay, now finally, what is replication? Doctors, why is replication the gold standard for medical research? How is this achieved with space research? Well, what does replication mean? Why is it so important?
So replication is so important because, we've got, more than a billion bases in each cell of the DNA, and we need to replicate that DNA to the highest fidelity possible. That process is a very complex process in the cell. It occurs in the nucleus. And when you get DNA damage is the precursor, the mutations that occur in genes are the precursors for cancer. And replication is a very sensitive component of the cell cycle where DNA damage can take place under stress, and you end up with mutations being encumbered into the genome. So that's replication.
So this is my wife giving me the honey-do list, and then I have to do it correctly, is what you're saying to me. Because if I don't do it correctly, I get in trouble. And in this case, a lot of radiation can produce a lot of trouble. All right. If you have questions about other terms you hear today or just general COVID questions, send those to us on YouTube, Facebook, Twitter, or the Medical News Network. The links are on your screen. Wait a minute, I got to make my phone quiet. For some reason, it wants to talk to me today. Let's shut that up. Okay. Nothing like modern technology to interfere with modern technology. Okay, Dr. Dana Hawkinson, Medical Director of Infection Prevention, Control, and Infectious Disease Specialist, is in the house now. He joins us with a COVID count for today.
Yes.
Doc Hawk.
Yeah, hi. I think it's going pretty good, Steve. We have 10 active infections right now in the hospital, 2 in the ICU, but none on the ventilator. Still 9 in that recovery period, but just as you and I were talking about, looking at the Washington Post cases and hospitalizations that they're able to present, looks like nationally case counts are going down, hospitalizations are trending down as well, so I think that's a good thing. A little bit trending up in Missouri from their data and a little bit trending down in Kansas. But
yeah, it is actually good news. And
Yeah.
just to say, 19 patients back in the day, we used to thought, oh, that was a lot. And then we got up to 225, 240 patients. We're like, okay, 19 is like a
Yeah.
walk in the park. But for if you're sick with COVID, it's still not a walk in the park. And long COVID is still very real. The signals out there that I'm seeing, Northeast cases have actually flattened and started to go down. Far West cases starting to flatten out. We're about 60 to 70% of the cases are the BA2.1, 2.1. There's some other BA2 numbers out there, and there's a few of the original Omicron, none of the Delta out there anymore.
Yeah.
So it's kind of this mismatch of different viral forms, but by and large, encouraging news here in the U.S., I think.
yeah, I would totally agree with you. I think that's right. Also looking at covariance.org, it looks like that we have a smattering or a smidge of BA4 in the United States as well. So, but overall, just very encouraging news. The BA.2.1, 2.1 still seems like it's the more predominant one at this point in time. But I think we are all optimistic in believing that hospitalizations and death will not match that peak that it did most recently with the Delta and Omicron surge. Hopefully, overall, things will start to flatten out as far as the number of people coming to the hospital. Hopefully because of vaccination and then previous infection immunity, but also testing early and getting on Paxlovid.
Yep, BA this, BA that.
Yeah.
It's everywhere you want to
Yep.
be. All right, do we have any reporter questions so far today? All right, hearing none, we're going to go back to our guest from Moffitt today. Doctors, tell us about your recent experiment you sent to the International Space Station as part of Axim Space's AX-1 mission. Dr. Duckett, you go first.
So this started when we were looking at the receptors that Patsy described earlier. These cell surface receptors, when they receive a signal, and they activate the beta-arrestin-1 pathway that we also just introduced. We elucidated a mechanism underlying this process. So when the receptors are activated by stress, so adrenaline, other catecholamines, for example, it activates this beta-arrestin pathway that results in a process that we have elucidated to now understand generates DNA damage. So chronic stress, chronic stimulation of this receptor ends up causing DNA damage. And when we, silence the beta-arrestin-1 gene in cells or in animals, we mitigate that DNA damage. And therefore, that was part of the experiment to find out whether the space travel with its stressful components, microgravity and ionizing radiation that occurs in the International Space Station, to see whether the beta-arrestin-1 silencing of the gene would mitigate the negative effects of the chronic stress that's been stimulated.
And so did that work? Because those guys are up in space for sometimes a very long time on the space station, kind of where I want to be. Go ahead. Did it work? Did the research prove?
So we're still working on that. So we sent 2 batches of cells to the ISS. One batch that expresses the beta-arrestin protein and the same cell type, but now is lacking the beta-arrestin protein. And they were sent on a microchip, so lab on a chip, that was inside a lab in a box, which was about the size of a shoebox, in fact. So the whole experiment was fully automated, where we were able to feed the cells, to keep the cells alive and happy, and maintain ambient temperature for these cells as well. So they spent, they were supposed to spend 8 days in space. They actually spent a lot longer there because there were some issues with the splashdown or the astronauts coming back just due to weather. So they actually stayed up in space longer than we anticipated. However, when it was returned to Earth and then returned to us, we were able to harvest the cells, and we now have these cells stored at -80 while we are preparing the control experiments that we have been performing here on Earth. So we wanted to expose the same cell types, those cells containing beta-arrestin and those lacking beta-arrestin, to different stresses here on Earth, so that we could compare those stresses to the microgravity and ionizing radiation stresses that the cells experienced when they were on the ISS. So the next step now is to extract the RNA, so genetic material, from all of these different experiments that we've done, so we can compare the gene expression profiles and see how they've changed following exposure to these different stresses. So the experiment's not complete yet, but we're hoping to have some answers in the very near future.
So here's what I really want to know. For all these folks flying around on the Enterprise, Discovery, Voyager, Stargazer, all these different ships that are really just important to our future, are they going to be able to actually do that in space? There's an awful lot of radiation out there, right?
Be protected from the ionizing radiation?
Yeah, is our body going to protect us, or do you think we'll be able to protect ourselves from all this ionizing radiation that's out there?
So what we're hoping we will see is that by ablating or silencing the beta-arrestin gene, we've obviously done this genetically where we've been able to silence the gene. What we would hope to do in the future, the long-term goal, is to generate a drug that mimics the silencing of beta-arrestin. And the idea would be that an astronaut, for instance, or any space traveler, whether they would be a commercial traveler, would be able to take a drug prophylactically, so ahead of time, and hopefully have that protection now on board or in the body, obviously, to protect the body from the DNA damaging effects of the ionizing radiation that they'll be exposed to.
Well, that sounds pretty cool. I like that. So the ultimate goal here on Earth is to prevent cancer. What are we ultimately hoping to achieve? Medication, vaccine, treatment? I mean, what's the link between what you're doing here and being at Moffitt Cancer Center? This is a great cancer program there.
Yeah, so we're understanding the molecular pathways that mediate the stress, the negative response to stress, which is the DNA damage. And by understanding that pathway, we're hoping to block the pathway or interfere with the pathway very specifically, not just by genetically knocking down beta-arrestin-1 or ablating beta-arrestin-1 overall function, but seeing if we can target the very pathway that leads to the DNA damage, and therefore mitigate the effects of persistent chronic stress, both in space and here on Earth.
Which would be amazing. So the previous studies that have been done about cancer cells, having been cultured in microgravity environments, has been done. How does that work apply to the cancer research? What does that work with microgravity? Tell us.
So cancer cells behave quite differently in microgravity. They will, when we culture cancer cells in the lab, for instance, we tend to grow them in a monolayer or in a 2D dish where the cells just lie very flat. And of course, in a tumor, the cells form spheroids, and this is how obviously the tumor gets its shape. And in space, on the ISS, what they found if they grow cancer cells there, that they will automatically form these spheroids. So they behave much more like a cell would within the context of a tumor under conditions of microgravity than they do when we culture them here in the lab. So it gives us a better understanding of how they would naturally form in the body. And the ISS is obviously a very unique environment, a very unique lab situation where we can study cancer cells in that way.
Does this mean that we should use extra gravity to treat cancer to keep them to going these funny shapes? That was a joke. I don't know. I'm just. All right.
there are some thoughts along those lines that microgravity could be beneficial, actually.
Isn't that interesting? I mean, it's fascinating to think the difference of space and when you're on a planet, obviously, and protected, because I think the atmosphere protects us from a lot of this bad ionizing radiation. So, all right. The desire to one day colonize in space drives a lot of interest by NASA, and yours truly, and researchers to determine if humans and animals could achieve multi-generational survival on the moon, Mars, or on other planets? And the answer, of course, is going to be yes. We just have to figure out how to do it. This is video from an experiment sent to the International Space Station by a colleague of our next guest. Dr. Joseph Tash is an emeritus professor in the Department of Molecular and Integrative Physiology in the KU School of Medicine. He reviewed and approved the astronauts' tests and procedures in using a special microscope and custom-made three-dimensional and gas-permeable slides, okay, that's a lot right there, slides to examine frozen samples of both human and bull sperm in the microgravity environment of the International Space Station. You know, why would we care about that? Actually, there's some pretty cool stuff around there. We're going to talk about that. The research showed the sperm could swim and move about normally. We didn't know that that was going to be the case. The studies have also showed that the sperm preferred to swim along surfaces which had not been seen before due to limitations caused by the gravity here on Earth. This finding may have future implications for couples who are infertile. That is also a benefit of environments in space and for implications for us here on the ground. Joining us again, Dr. Lane Christensen. You are conducting your own research on the effects of space on humans. Tell us about your work around galactic cosmic radiation, which happens in deep space.
Okay, so I first want to just preference that this work is a team approach, much like Dr. Duckett and McDonald. This is led by a group at the Ames National, or the NASA Ames Research Laboratory in Mount View, California, and they have used the Brookhaven National Laboratory Linear Accelerator to actually expose animals, and most of our work is done with mice, to galactic cosmic radiation. These are not something that we see on Earth. And
that's what that linear accelerator in Brookhaven does, right?
Yeah,
it acts like global cosmic radiation.
Right.
That's pretty cool.
Yeah.
Okay.
And the other aspect of it is, even in on the International Space Station, when we look at the amount of ionizing radiation that's present there, it's much lower because it's inside of the Van Allen belt
around the Earth,
which protects us from that GCR. So we don't actually see very much GCR.
Which
radiation.
is a good thing, because GCR is sort of bad for the human body.
It's very, very bad.
Yeah.
And when you go to the moon, though, however, or to Mars, which is the goal, or if you're a Star Trek fan,
yeah.
you want to be in deep space.
Deep space, yeah. Warp me out. Let's do
You're
it.
going to have you're going to be exposed to a lot higher levels of galactic cosmic radiation that the present we don't have good shielding mechanisms for. So there's because those aspects are not very well shielded by the common things that block radiation on the Earth.
But the titanium hull within within Enterprise should be due to the blocker, I think. Is that the point?
I don't know about that.
Okay, well, it sure is on Star Trek. All right. All right, well, that is pretty cool to study this stuff. But what's the ultimate goal of what you're trying to establish with this?
Okay, so that work is with the galactic cosmic radiation studies are actually in preparation for those flights to Mars, where we send astronauts into deep space for a long period. It's estimated it'll take about 3 years to go to Mars and come back, which is a large and long exposure to GCR. The other work that we do is looking at this effects on the female reproductive system, and looking at the ability of the female reproductive system to work in space, as it's very important in the development of or maintenance of the bones and the metabolism in female astronauts.
This is pretty remarkable stuff, and the kind of thing that I actually get the biggest kick out of. So how important are these kinds of studies in the big picture for fighting disease like cancer and helping us to try and be healthier here on Earth, do you think?
Well, I think that one of the things that I think we learn from these type of studies is, going into another atmosphere or into another environment. It tells us a lot about our own environment. I mean, everything is, we've evolved from millennia to be present on Earth under this 1G under gravitational force. When we send people into space, all of the systems have to relearn how to work. We know there's effects on the cardiovascular system, massive effects there. When the astronauts come back down, you've seen them; they always get helped off of the platforms because their muscles have been weakened, their bones are weaker, and a lot of that has to do with the hormones in the body that regulate that aspect. And some of those hormones are coming from the reproductive system, which we are studying.
Yeah, that is pretty amazing. And again, why the reproductive system, though? You were telling me a little bit about the show why that's so true, why it's important to look at the reproductive system of all parts of the body.
Well, so the reproductive system is making it makes the estrogen that's necessary for bone development and growth, and as well as it's very important in making, obviously, in the male side, the testosterone, which ultimately will be converted to estrogen, which will have its impacts on the bone. So even in the the male side, those the testis and the gonads are critical involved in the.
So when we talked a little bit on the introduction just a moment ago, it was like talking about how sperm like to swim along surfaces. Mm-hmm. And so does that mean they're still going to work and that a man and woman could reproduce in space?
Okay, so I knew this question would come up.
Yeah,
So it's one of those so far we don't know. And right now our work is focused on trying to understand if on the female side, does the ovulation occur? And does the oocyte, once it gets into the fallopian tube, is it able to be moved to the site where fertilization would occur? It's an unknown at this point. But the fact that the sperm actually, in that video, progress along the surface is probably not. Is probably very similar to what happens on the ground. We just don't know that.
Wow, that is amazing stuff. So how important are these kinds of studies, do you think, for fighting disease like cancer and helping us stay healthy here on Earth? And I'll take that for you. Yeah, Doctor, anybody.
So I think Dr. Christensen just touched on some excellent points there. We're looking at how astronauts' bodies respond to their time in space, where you see effects on the cardiovascular system, bone and muscle wasting. And by studying how these processes occur in these astronauts, could potentially lead to developing drugs that can help patients and people on Earth. If you think about muscle wasting, for instance, this is something that we see frequently in certain cancers, pancreatic cancer, for instance, where there's a high prevalence of cancer-associated cachexia, muscle wasting, weight loss, etc. So by being able to study these processes and how they occur in space, we do know that the astronauts can recover when they return to Earth, but that's not the case with patients who are suffering from cachexia. So there's a lot to be learned on how the body reacts to being in space and how it recovers when the astronauts return to Earth. So I think there's a lot to be learned here that can be applied to diseases like cancer.
Yeah, that is fascinating. So really, what I was always appreciating in Star Trek is when they had a medical malady, first of all, they always did worry about radiation, and Dr. McCoy was always zapping people in the arm so that they could be protected against radiation. I must have been that beta-arrestin 1 thing he was taking care of. And then, and I'm always impressed by how fast the antidotes could be developed. So I'm really expecting some pretty quick results here from y'all's experiment and y'all's work, right? Isn't that sort of how this, because they did it within a 60-minute episode, people. That's what I'm saying here. Okay. Alexis, you've been monitoring questions from our community. What are folks asking about so far today?
Dr. Stites, I am getting so many questions from people. Let's start with Norman. Norman wants to know, how long does COVID stay in your system? Can you still test positive but be contagious and shedding the virus?
Dr. Hawk?
Yeah, as far as, what does that mean to stay in your system? I'm wondering if this means just in the upper respiratory tract where you're testing. Certainly we know that the virus can get into your blood transiently for a very short period of time, but I believe probably the question here is in the upper respiratory tract where you are testing. We know that overall the viral stage of the disease should really be over by about 10 days after symptom onset. You really shouldn't have very much viral replication at that point. However, we know that you can still test positive after that because of viral remnants or genetic material remnants, either by PCR testing or antigen testing, but we also do understand that more than likely the antigen testing is continuing to be evaluated as more of a sign of active replication of the virus. So, but overall, we know that you can continue to test positive even after symptoms subside, especially with PCR tests. So, I don't know if that answers your question. We know PCR tests can be positive for 30, 60 days even. The antigen test should be not as often positive, especially if you are not in that infectious viral stage.
Hawk, the only thing I would add to that is there are some viruses that can hide out in your body, like herpes virus and CMV. They can exist in nerve endings and reactivate. We don't think COVID or SARS-CoV-2 does that. Long COVID is really not long amounts of virus. It's a reaction your body develops to the virus, and so there are viruses that can sit in your body for a very long time, for years, for all your life. If you get herpes virus, that's why you can get cold sores. They come and go again and again, and shingles, etc. And then other things like SARS-CoV-2 seem to be eliminated relatively fast.
Exactly. I would say, with things like chickenpox, which becomes shingles, or herpes viruses, they actually integrate into the cellular DNA. This does not happen with SARS-CoV-2. We understand there may be some still viral remnants going on, and a very low level of replication possibly. We are looking into long COVID about that, for instance, in the gut or some of their organs. But overall, just like you said, there isn't that active replication, and it should not, and it does not integrate into your genome, into the DNA. So.
Alexis.
Isaac has another question. I think Isaac is looking for some clarification. He writes, are cases going down, or has the current wave peaked? Are prior infections and vaccines keeping the numbers down, or simple lack of data due to home tests?
Okay, well, that's a good question.
Yeah.
There's multiple answers there. The wave is a funny one because it's already escalated in the Northeast and come back down. The Midwest, and also to some degree in the West. I think the Midwest, Hawk, has really, we don't know if it's peaked or not. One of the questions that was part of that, we don't really ever will know the full numbers, and that's why the CDC moved from looking at total numbers of tests being done and how many of those were positive to hospitalizations as the bigger sense of what a wave is. There was a mild rise in hospitalizations, nothing like it had been in the Delta or the original Omicron, but there were still a lot of cases, just as not as many hospitalizations. And I think what that tells us, Hawk, is that really prior infection and especially vaccination has helped blunt this wave.
Yeah, I certainly believe that, and we've talked about that. The individual immunity you have, again, the safest way to get that is through vaccination. Certainly people have been infected and reinfected. Just was reading an article today, Lancet Infectious Diseases, which did continue to support the fact that if you are infected, your chance of having a reduced chance of hospitalization and mortality is improved if you do get vaccinated, even after infection. But overall, I don't think we're going to know. To answer Isaac's first question, we need to follow this 7 to 10 day trend. This was really the first day that I saw a downtrend in number of cases and hospitalizations. It may have occurred yesterday. But again, we've had the long holiday weekend, so we don't know all the full data. But hopefully, moving forward to the next 7 to 10 days, we will continue to see that arrow trend going down as far as cases and especially hospitalization.
It's actually been estimated that the total number of cases are as high as the Delta wave. But we don't have that information because there are so many home tests and so many fewer publicly reported tests being done. And as a result, we've underestimated the total size of the wave. But many people say that this wave is as big as the Delta wave in terms of the number of patients who are infected, the number of folks who are infected. The number of hospitalizations is significantly less.
Yeah, absolutely.
And I do think that's because of prior immunization and prior infection. And hopefully that will hold us for a while.
Yeah.
And we're very happy that the number of deaths have not skyrocketed as they have in the past.
Yep.
Alexis.
I have another question from Archie. Archie writes, my husband just tested positive last night. He's vaccinated and boosted. It's like a head cold. He got Paxlovid yesterday. How does Paxlovid help? Is it easy to get for most people, and who should take it?
Oh, great questions. We know there's a lot more Paxlovid out there now, Hawkeye.
Yeah.
In fact, I think people are being encouraged to try and get it because there's, I don't want to say there's a surplus, but there's plenty of Paxlovid available. So I think if you meet the criteria, and the criteria are fairly wide, and Hawk, we can cover that.
Yeah.
I think that if you meet the criteria, my sense is, and you don't have to worry about the drug interactions, because that's the one hang up with Paxlovid, is there are a lot of drug interactions. You have to talk to the pharmacist and your primary care doc to make sure that you're safe. But if those things are true, Paxlovid reduces hospitalizations by 85%. That's a really big number.
Yeah, absolutely. If you are vaccinated and up to date with vaccination, you have an 80% or more reduction in chance of hospitalization. If you take Paxlovid, on top of that vaccine protection, you have a 90% reduction in going to the hospital as well. Just as you said, it is a very broad definition of who should get it. It is recommended for people at high risk of progression to hospitalization. What is high risk? Well, you have to go to the CDC, and just like you said, Steve, there are a number of different comorbidities in there, such as weight, diabetes, heart disease, lung disease, just a bunch of chronic conditions. So, but overall, yeah, it will reduce your chance of hospitalization. What it will do, and it is most effective in those first 5 days after symptom onset, will reduce that viral replication stage so that your body's immune system can then hopefully clear that virus even more quickly with the assistance of this medicine.
And I would just say, I think if you and your doctor think, okay, this is the right thing to do, I wouldn't hesitate. Like, I'm 62 years old, I have a little hyperlipidemia, a history of prostate cancer. You know what? If I get COVID, I'm going to take Paxlovid. I'm fine, bring it on, because the drug's remarkably safe. It is remarkably effective, and I think it's something people should seriously entertain if they have it. Just talk to your primary care doc and get that figured out.
Yeah, I would certainly agree. I think if you're over 60, you probably should go ahead and take it. If you're in that 50 to 60 range, talk with your physician. If you have other comorbidities, certainly get it. Do not be alarmed at this point in time if you have rebound of symptoms in 5 to 7 days after completing the course. Remember, it is a 5-day course. Please complete the whole course. But if you do have a rebound in symptoms, remember you have to go back to isolation again for the 5 days, just as you would when you initially got it. But they haven't seen any more severe disease or hospitalizations in those people that have had rebound, which is only about 2% in the clinical trials. The other issue is we have not currently seen any resistance to Paxlovid, which is a very good thing as well.
We like that. Okay, Alexis.
I have another question from Norman. Norman would like to know, I guess Norman wants you to look into your crystal balls and tell us, do you think we're headed towards another mask mandate and or another lockdown? Children's Mercy, Norman writes, moved back into the yellow yesterday.
Yeah, I think that had to do with Jackson County and some other factors. I'll be surprised right now. I would be surprised if we go to a mask mandate or a lockdown. I think if you are immunosuppressed and you haven't been vaccinated, or you're not sure you respond well to vaccination, I think you should be masking wherever you go, because there is a lot of community spread of the BA.2.1.2.1 variant. And so I think for people like that who are at risk, you should put a mask on. As far as a broad mask mandate or community lockdown, I don't see that on the horizon right now. And the only way that will come about is if there's a really severe strain that develops. And I think we've learned so much more. We have such better tools with Paxlovid and some of the other drugs, Hawkeye. I'm hopeful we can avoid those types of really extreme measures that we had to take on at first when we had a population who had not had it.
Yeah, absolutely. I think it's very difficult. We know from a public health and a medical standpoint, the masks are always going to be the best option. We know when you are a legislature or a community leader making those decisions, there's much more details that go into making that decision. Steve, you started this conversation last night on email about our facility, and we know, I think we have to consider the public realm as different than the health systems or a medical care facility, just because there is such a high concentration of those that are at very high risk of progression to disease. So I think that's something different than a healthcare facility, the general public is. But I would agree with you. I think it's going to be difficult to institute that.
Yeah, and I think the key will be is if you start seeing hospitals under a lot of stress again, like we did last December. That's a different story. Then we need the public's help to keep all of us safe from all diseases, not just COVID. Because when we were that busy, we couldn't take care of strokes and heart attacks. So that put everyone at risk, and that's when we really needed the help of the public to try and make sure we could get through that crisis together. Right now, I think hospitals have capacity. I think we're okay.
Kathy would like to know, are we now in an endemic officially, or is this just wishful thinking?
Well, I think, I don't know, we're somewhere between endemic and pandemic, and it's just going to be around for a while. So, Hawkeye, I think we're going to be living with it, and I think we have to be ready to adapt and be remember we said before, you got to be nimble at this point.
Yeah.
So when things start to rise, or we get in trouble, be ready to do those public health measures, because we said before, the rules of infection control, they travel with you wherever you go, they keep you safe.
Yeah, I agree with that too. I think it's all about semantics at this point. Also understand our country's experience as opposed to other countries. So the pandemic, if you will, what we were used to for the last 2.5 years is probably over for the United States at this point in time. It is endemic. We know around the world it is endemic. It is circulating all the time. But what about a pandemic? Well, we know China has had recent surges and lockdowns. We know North Korea. We know the experience of other countries is quite different, and that can affect things like supply chain. And so as far as the rest of the world goes, I believe there is still a pandemic because we still have these disruptions in so many aspects of life for everybody. But in the United States, it is endemic. It continues to circulate. It is going to be and continues to be extremely important for you to protect yourself individually, and that is by making sure that you and your bubble or your family are vaccinated and up to date with vaccines.
Yeah, I think your point about it's a play on words at this point. The definition of pandemic and endemic is so loose and it's so gray that the reality is you got circulating virus. We're going to have to be nimble. Things get worse again. We got to take more public health measures. When things are better, we don't. We've got better therapies, and if you just get vaccinated, you can really help control this and not see the wild spread. And remember, vaccination is not evil, and it doesn't cause you to grow 3 eyes and 4 horns.
I have a question from Anthony. This involves outer space.
Awesome.
Anthony would like to know, can COVID survive in outer space?
Oh, there we go. So do viruses survive in outer space? Team, let's take that on. I got nothing here, so you're going to have to help us out. Anybody?
Who's?
Yeah, it's a great question, and don't know too much about viruses. We do know that some bacteria develop resistance to antibiotics that they would ordinarily be susceptible to here on Earth. So there's certainly the possibility that viruses can develop resistance, I would say, in space, but I don't know that those studies have been done.
Okay.
I think we just need to go get some of that global cosmic radiation, Dr. Lane. Let's get. Look, here we are. Can we give it to them like that?
I would echo that. I don't know that we know the answer to that problem of viruses. Are they able to? But one of the things we do when we send astronauts into space is they're locked down for a good period of time, so they don't take things into space, and that's very important.
And if I remember, all the cultures on the moon didn't produce any bacterial or viral growth, right? So maybe they didn't survive, and there's a fair amount of different kind of funny radiation. There's not a lot of atmosphere around the moon. So yep. All right.
Okay. I've got one
We solved that crisis quite.
I got a question from Julie. Julie would like to know a question. This is specific to long COVID. Have markers or pre-existing conditions been identified that show an increased chance of having long COVID?
Hawkeye, there were some, but I think that they're not very strong signals.
Yeah, I would agree. I think there have been some genetics that have been identified. As far as severe illness for long COVID, I don't recall off the top of my head, but again, when we are talking about genetics, it isn't just one gene as we learned about in school when Mendel was going through pea plants and flowers as far as genes like that. It is a complex interplay of many factors in our body. And certainly we have, again, I think we've seen this and understood this for severe disease. For long COVID, I don't really know that we have any specific genetic factors that we've identified at this point, but could be wrong.
One more question from Angela. Angela would like to know if you can take another round of Paxlovid if your symptoms come back.
I don't believe that another round of Paxlovid is recommended at this point in time. It is just to continue to isolate and treat those symptoms symptomatically as needed with, again, hydration, Tylenol, and things like that. I haven't seen any recommendations for a second course of Paxlovid.
Alexis, you're muted there. Go ahead. What you said?
Okay, anxious mama. She would like to know, Melissa would like to know, any update on the under 5 vaccine?
I believe that ACIP, the Committee on Immunization Practices, and the FDA, at least ACIP I know, is meeting next week. I think June 8th was their first meeting. They have 3 meetings in fairly quick succession, so we should have more information. I think we were still believing that 3rd or 4th week of June, possibly, it may be recommended. So that's the latest thing I know, but I think their first meeting was June 8th to go over all the data.
We'll find out soon how that goes. Okay, so I have a question, and then we need a final comment from all of our guests today. First of all, I love space, the final frontier. These have been the voyages of the Morning Medical Update, going where no man has gone before, or no person has gone before. So thanks for being a part of that program. So tell us what you think the biggest obstacle is to space travel for man to go forward. So I'm going to start with Dr. Duckett and Dr. McDonald. What are the biggest obstacles you see out there?
So I think, certainly one of the biggest obstacles is going to be the galactic radiation that you're exposed to when you get beyond, into outer space, and mitigating the effects of the DNA damage and other damage that occurs with galactic cosmic radiation is going to be a major factor to overcome.
Okay, Dr. McDonald.
Yeah, I think obviously, the longer and further space travel becomes, we're going to be faced with other questions that we need to answer. Again, it's the duration of the flight, how far they go into space. What effects are we going to see that might be different to what we're seeing now on the human body when astronauts and space travelers are exposed to, more chronic exposure to the stresses that they experience now. Again, what are the long-term effects likely to be once they return to Earth?
Yeah, that's really cool. Dr. Christensen.
So I'm going to echo both Dr. Duckett and McDonald in the context that we don't really have the long-term studies of galactic radiation as well as microgravity that only can be replicated really in space. We can do either or, but we haven't been able to do those experiments and to really understand what's going on. I think those are aspects that will be a big player in when we get to that next level, and hopefully soon.
It's fascinating because our bodies are really built for this environment and this gravity and these bacteria and viruses. And space is a whole different environment with lots of different kinds of environments out there. All right. Any final thoughts, Dr. Christensen? Dr. Christensen, sorry. I'm pointing around.
No, I'm good.
All right. Dr. Duckett, Dr. McDonald, final thoughts for today?
Well, we'd just like to say thank you for inviting us to be part of this experience. And we've certainly learned a lot. And so we'll keep you posted on how these experiments turn out.
All right.
It's great fun. Thank you.
All right. Well, thanks to you guys. This is awesome. We're going to have you back because, I love space. Hawkeye, final thoughts?
Yeah, Thank you to our guests. Such an interesting program today. We hope to learn more. And I think as we are putting more and more people into space overall, hopefully the research can expand as well, because I think it's vitally needed. So, good.
It's funny. It reminds me that the journey of humankind is one of trying to build the airplane while we fly it. And whether we're talking about outer space or we're talking about COVID, we learn a lot along the journey. But aren't we at our best when we're trying to take that journey together and not trying to fight about some of the harder things, not the harder, but some of the different things in our daily lives? And when we can look forward and we can look up, think of what we can do. Thanks for joining us. Join us tomorrow for another morning medical update. I'll be back tomorrow morning as Jess is out. Not Alexis, the Jess is out, and I'll be back and here, and we look forward to seeing you then. Thank you so much for joining us today. We'll see you soon.
Summer is here. Many will be traveling or spending time in the sun and at the pool. I'm Jessica Lovell. What you can do to keep your skin cancer-free and your kids safe in the water. We'll see you Friday at 8 on these social media channels.
Subscribe to our morning medical update and Open Mics with Dr. Stites podcast. Now, everywhere podcasts are available.